ค่ะกราบสวัสดีนะคะท่านอธิบดีกรมวิทยาศาสตร์การแพทย์ท่านรองอธิบดีท่านผู้ทรงคุณวุฒิท่านวิทยากรท่านคณะอาจารย์จากศิริราชและผู้มีเกียรติทุกท่านค่ะขอต้อนรับทุกท่านนะคะเข้าสู่การสัมมนาวิชาการวิทยาศาสตร์การแพทย์ครั้งที่1ประจำปี2566ในวันนี้ค่ะดิฉันนางสาวพินิมิตรแดงไพโรจน์และดิฉันนางสาวกัญญากรนำจันทรารับหน้าที่เป็นพิธีกรในวันนี้ค่ะ
Good morning, ladies and gentlemen. Welcome to our first 2023 the Medical Science Seminar on the topic of gene therapy. Today, I'm Ganyakorn Namjantra and
I'm Pinimit Dang Pairod.
Be the hosts for today.
ค่ะและในวันนี้นะคะเนื่องจากท่านอธิบดีติดภารกิจสำคัญท่านจึงอัดคลิปวิดีโอในการเปิดการสัมมนาในวันนี้ค่ะ
As today, Dr. Supakit Silila, Director of the Department of Medical Science, couldn't be here today, so he has sent his gratitude and his opening speech for today's opening ceremony as a video.
ค่ะในโอกาสนี้นะคะขอกราบเรียนเชิญท่านหัวหน้าสำนักวิชาการวิทยาศาสตร์การแพทย์ท่านดร.ประภัยวงศ์สินคงมั่นกล่าวรายงานการสัมมนาวิชาการวิทยาศาสตร์การแพทย์ครั้งที่1ค่ะ
On this occasion, I would like to invite Dr. Prapai Wongsin Kongman, Head of Medical Science Technical Office, to deliver the report. Please welcome.
เรียนท่านอธิบดีดิฉันขอกราบขอบพระคุณท่านอธิบดีเป็นอย่างสูงที่ได้กรุณาให้เกียรติมาเป็นประธานในพิธีเปิดการสัมมนาวิชาการวิทยาศาสตร์การแพทย์ครั้งที่1ประจำปีงบประมาณพ.ศ. 2566ในครั้งนี้ตามที่กรมวิทยาศาสตร์การแพทย์โดยนักวิชาการของกรมวิทยาศาสตร์การแพทย์ได้เข้าร่วมในการประชุมครั้งนี้โดยผู้จัดประกอบด้วยสำนักวิชาการวิทยาศาสตร์การแพทย์สถาบันชีววัตถุสถาบันชีววิทยาศาสตร์ทางการแพทย์สำนักยาและวัตถุเสพติดและกองแผนงานและวิชาการได้มีความร่วมมือกันในการวิจัยในด้านเซลล์ต้นกำเนิดและผลิตภัณฑ์การแพทย์ขั้นสูงร่วมกับทางคณะแพทยศาสตร์ศิริราชพยาบาลและสถาบันชีววิทยาศาสตร์โมเลกุลมหาวิทยาลัยมหิดลในการพัฒนาผลิตภัณฑ์เซลล์ต้นกำเนิดชนิดมีเซนไคมอลสเต็มเซลล์มานานกว่า10ปีและได้ขยายสู่การวิจัยและพัฒนาสู่ผลิตภัณฑ์ยีนบำบัดและผลิตภัณฑ์เอ็กซ์ตราเซลลูลาร์เวสเคิลจากมีเซนไคมอลสเต็มเซลล์ดังนั้นในปี 2566 นี้นับเป็นโอกาสอันดีที่ท่านศาสตราจารย์แพทย์หญิงละอองศรีอัจฉริยสกุลหัวหน้าโครงการวิจัยได้จัดให้มีการประชุม Research Team Meeting 2023 โดยได้เชิญดร. Timothy Stout และดร. Mark Pennesi มาให้ความรู้และแลกเปลี่ยนประสบการณ์กับนักวิจัยชาวไทยในการจัดสัมมนาวิชาการในครั้งนี้ได้จัดเป็น2รูปแบบคือการจัดสัมมนาในห้องประชุม110และการจัดสัมมนาในรูปแบบออนไลน์ด้วยโปรแกรม Zoom และ Facebook Live สำนักวิชาการวิทยาศาสตร์การแพทย์ให้แก่บุคลากรส่วนกลางศูนย์วิทยาศาสตร์การแพทย์และผู้สนใจในการสัมมนาวิชาการทั้งนี้มีผู้สมัครเข้าร่วมการสัมมนารวมทั้งสิ้น132คนวันนี้ดิฉันขอเรียนเชิญท่านอธิบดีกล่าวเปิดการสัมมนาวิชาการวิทยาศาสตร์การแพทย์ครั้งที่1เพื่อเป็นเกียรติแก่ผู้เข้ารับการสัมมนาและท่านวิทยากรในการบรรยายครั้งนี้ต่อไปขอกราบเรียนเชิญค่ะ
ท่านรองอธิบดีท่านวิทยากรท่านผู้ทรงคุณวุฒิท่านผู้ในการผู้บริหารแล้วก็ผู้เข้าร่วมการสัมมนาที่เคารพรักทุกท่านครับก็วันนี้เป็นวันที่ดีอีกวันหนึ่งนะครับผมมีความรู้สึกยินดีนะครับที่ได้มาเป็นประธานในการเปิดสัมมนาวิชาการวิทยาศาสตร์การแพทย์ครั้งที่1นะครับแล้วก็ขอต้อนรับอาจารย์วิทยากรที่สำคัญนะครับคือศาสตราจารย์แพทย์หญิงละอองศรีอัจฉริยสกุลดร. Timothy Stout และดร. Mark Pennesi ที่ให้เกียรติมาบรรยายในเวลานี้นะครับต้องเรียนอย่างนี้ครับว่ากรมวิทยาศาสตร์การแพทย์เนี่ยเป็นหน่วยงานของกระทรวงสาธารณสุขที่เราทำหน้าที่ ในการศึกษาวิจัยวิเคราะห์ทั้งหลายเพื่อให้เกิดองค์ความรู้ในด้านวิทยาศาสตร์การแพทย์ที่จะตอบสนองเรื่องของการวินิจฉัยโรครักษาพยาบาลรวมจนถึงเรื่องของการคุ้มครองผู้บริโภคของประชาชนในชุมชนด้วยอันนี้เป็นเรื่องสำคัญที่เราเป็นจิ๊กซอว์ส่วนหนึ่งในการแก้ปัญหาสาธารณสุขของประเทศนะครับนอกจากนั้นเนี่ยถ้าทุกคนได้ติดตามข่าวสารเนี่ยวันนี้เนี่ยประเทศไทยเรากำลังเดินหน้า policy ที่เราเรียกว่า BCG model นะครับส่วนหนึ่งที่มีความสำคัญก็คือเรื่องของการวิจัยและพัฒนาผลิตภัณฑ์ทางการแพทย์ชั้นสูงที่เราเรียกว่า ATMP Advanced Therapeutic Medical Product เนี่ยนะครับสิ่งเหล่านี้จะเกิดขึ้นไม่ได้ถ้าไม่มีองค์ความรู้ที่สำคัญจากการวิจัยพัฒนานะครับแล้วก็สุดท้ายต้องผลักดันให้เกิดผลิตภัณฑ์อย่างที่ว่าแล้วก็ใช้ประโยชน์ได้จริงนะครับพูดง่ายๆว่าจากห้องแลบไปสู่โรงพยาบาลของการใช้งานจริงไม่ใช่จากห้องแลบแล้วก็ขึ้นหิ้งหรืออยู่บนเชลฟ์เท่านั้นเพราะฉะนั้นการจะทำอย่างนี้ได้เนี่ยเครือข่ายความร่วมมือของหลายๆฝ่ายเพราะเราพบว่างานวิจัยหลายงานเนี่ยนะครับมันต้องการองค์ความรู้ที่หลากหลายสนับสนุนต่อเนื่องกันนะครับเพราะฉะนั้นการที่มีเครือข่ายวิจัยต่างๆเนี่ยมาร่วมไม้ร่วมมือกันในประเทศเนี่ยโอกาสที่จะเกิดความสำเร็จก็จะมีมากขึ้นกรมวิทยาศาสตร์การแพทย์รวมถึงหน่วยวิชาการต่างๆอาทิเช่นเครือข่ายความร่วมมือกับคณะแพทยศาสตร์ศิริราชพยาบาลสถาบันชีววิทยาศาสตร์โมเลกุลของมหาวิทยาลัยมหิดลสำนักงานพัฒนาวิทยาศาสตร์และเทคโนโลยีแห่งชาติเนี่ยนะครับก็ได้ร่วมไม้ร่วมมือกันทำเรื่องพวกนี้มาต่อเนื่องยาวนานพอสมควรจากคำบรรยายของท่านหัวหน้าสำนักวิชาการวิทยาศาสตร์การแพทย์เนี่ยก็คิดว่าวันนี้เป็นโอกาสที่ดีมากครับที่เราได้วิทยากรที่มีความรู้ความเชี่ยวชาญนะครับในเรื่องของสิ่งที่เรากำลังจะเรียนรู้กันนะครับในด้านของยีนเทอราปีเนี่ยนะครับจากดร. ทิโมธี สตาวท์แล้วก็ดร. มาร์ค เพนเนซีซึ่งเป็นผู้ทรงคุณวุฒิในระดับโลกนะครับอันนี้เนี่ยนอกจากเราจะได้องค์ความรู้จากเขาเรายังจะได้ความร่วมไม้ร่วมมือหรือการสร้างเครือข่ายพันธมิตรในการทำงานเรื่องพวกนี้ต่อไปทั้งในปัจจุบันแล้วก็ในอนาคตนะครับอันนี้จะเป็นประโยชน์มากในการที่เวลาเราจะทำนวัตกรรมใดๆเนี่ยก็จะทำให้เกิดโอกาสมีความสำเร็จมากขึ้นได้แล้วก็เป็นความเข้มแข็งของเครือข่ายที่จะทำเรื่องอื่นๆต่อไปในอนาคตนะครับทีนี้ถ้าเราพูดถึงเรื่องยีนบำบัดนะครับหรือยีนเทอราปีเนี่ยนะครับรวมถึงเรื่องของผลิตภัณฑ์การแพทย์ขั้นสูงเนี่ยจริงๆประเทศไทยเราเนี่ยก็ทำมาหลายเรื่องนะครับมีผลงานบางเรื่องเนี่ยที่ก็เข้าใกล้ความเป็นจริงหรือบางเรื่องก็ได้นำไปสู่ขบวนการที่จะนำไปใช้จริงมากพอแล้วนะครับเพียงแต่ว่าการวิเคราะห์วิจัยทั้งหลายเนี่ยเราก็ต้องยอมรับว่าเรามาทีหลังชาติที่พัฒนาไปแล้วหลายประเทศนะครับเราก็ต้องตามให้ทันนะครับเพราะวันนี้เราอยู่ในระดับที่ทำการทดลองเสียเป็นส่วนใหญ่นะครับที่ออกมาเป็นโปรดักต์ที่ไปสู่มือผู้ใช้จริงๆอย่างเป็นรูปธรรมเนี่ยก็อาจจะยังมีไม่มากนะครับเราได้ร่วมไม้ร่วมมือจะยกตัวอย่างครับท่านผู้เข้าร่วมสัมมนาที่รักทุกท่านครับกรมวิทยาศาสตร์กับทีมวิจัยของศาสตราจารย์แพทย์หญิงรองศรีอัชนียสกุลเนี่ยนะครับอาจารย์รองศรีนะครับคณะแพทยศาสตร์ศิริราชพยาบาลเนี่ยได้ทำการเตรียมแล้วก็ตรวจสอบคุณสมบัติของเซลล์ต้นกำเนิดที่เราเรียกว่า mesenchymal stem cell นะครับซึ่งเรื่อง stem cell ก็เป็นอีกarea หนึ่งเป็นพื้นที่หนึ่งที่กำลังพูดถึงกันเยอะแล้วก็มีการพัฒนากันอย่างพูดง่ายรวดเร็วมากเซลล์ตัวนี้เนี่ยเราจะใช้ในการรักษาผู้ป่วยโรคตาครับเป็น retinitis pigmentosa ซึ่งขณะนี้เนี่ยผลงานในเบื้องต้นเนี่ยก็เป็นที่ยอมรับเพราะว่าได้มีการตีพิมพ์ในระดับวารสารซึ่งเป็นวารสารที่ได้รับการยอมรับเนี่ยนานาชาติในปี 2021 มาแล้วนะครับแล้วก็กำลังพัฒนาต่อยอดนะครับเพื่อที่จะทำการเตรียม extracellular vesicle นะครับจากเซลล์ต้นกำเนิด mesenchymal เนี่ยนะครับมันจะมีประโยชน์ในการที่จะใช้กันในการใช้งานและใช้ในการเก็บรักษาได้ดีกว่าของเดิมนะครับเพราะฉะนั้นอันนี้เนี่ยเป็นการพัฒนาต่อยอดไปอีกรวมถึงพัฒนายีนบำบัดสำหรับการรักษาโรคตาความสำคัญมีอย่างนี้ครับท่านผู้เข้าร่วมสัมมนาครับ วันนี้เนี่ยถ้าเราต้องการผลิตภัณฑ์ตัวนี้เนี่ยนะครับเราต้องไปซื้อหามาจากต่างประเทศนะครับค่าใช้จ่ายเนี่ยกว่า 400,000 เหรียญนะครับก็เอา30 กว่าบาทคูณเข้าไปเนี่ยนะครับต่อการรักษาตาเพียงหนึ่งข้างนะครับเพราะฉะนั้นมันยังแพงมากแล้วก็เป็นเรื่องที่ต้องซื้อหามาจากต่างประเทศนะครับเพราะฉะนั้นถ้าทีมวิจัยของเราที่นำโดยอาจารย์รองศรีกับความร่วมมือทั้งกับกรมวิทยาศาสตร์การแพทย์ด้วยเนี่ยถ้าเราทำเรื่องนี้สำเร็จนะครับเราก็จะทำให้เกิดพูดง่ายๆของที่มีคุณภาพดีแล้วก็ราคาที่อาจจะจับต้องได้มากขึ้นเพราะต้องเรียนทุกท่านครับว่าในชุดสิทธิประโยชน์ของหลักประกันใดๆกองทุนหลักประกันใดๆของประเทศเนี่ยนะครับก่อนที่จะเอามาใช้กับผู้คนอย่างกว้างขวางเนี่ยมันต้องมีการคิดเรื่องของความคุ้มค่านั่นหมายความว่าประโยชน์ที่เกิดขึ้นกับค่าใช้จ่ายที่เกิดขึ้นเนี่ยมันต้องมีความเป็นไปได้นะครับถ้ามันยังแพงมากอยู่เนี่ยมันก็จะได้ตัวเลขความคุ้มค่าออกมาน้อยนะครับซึ่งโอกาสที่จะทำให้คนไทยทั้งหลายเนี่ยได้เข้าถึงเรื่องพวกนี้ก็จะยิ่งห่างไกลไปอีกเพราะฉะนั้นถ้าเราทำได้เองในประเทศนะครับแล้วลดต้นทุนลงมาลดค่าใช้จ่ายลงมาไม่เป็นการเสียเงินตาไปจากต่างประเทศเนี่ยโอกาสที่พี่น้องคนไทยจะได้ใช้ประโยชน์จากเรื่องนี้ก็จะมีมากขึ้นนะครับอันนี้เป็นเรื่องที่มีความสำคัญแล้วก็สอดคล้องกับนโยบายที่เราพยายามทำเรื่อง BCG โมเดลนะครับที่จะขับเคลื่อนเรื่องพวกนี้ในด้านของยาเวชภัณฑ์พวกนี้ต่อไปในอนาคตนะครับสุดท้ายผมคงต้องขอบคุณศาสตราจารย์แพทย์หญิงละอองศรีนะครับอัจฉริยสกุลนะครับที่อาจารย์เป็นพูดง่ายๆว่าเป็นบุคคลที่สำคัญในการที่จะนำในวิชาการในเรื่องนี้นะครับรวมถึงกรมวิทยาศาสตร์การแพทย์ในส่วนที่เกี่ยวข้องนะครับและผมยังต้องขอบคุณอาจารย์วิทยากรที่จะมาแลกเปลี่ยนให้ความรู้กับเราในการสัมมนานี้คือดร. ทิโมธี สตาวแล้วก็ดร. มาร์ค เพนเนซีนะครับที่ท่านสละเวลาแล้วก็พูดง่ายๆตั้งใจในการที่จะให้ข้อมูลเหล่านี้กับพวกเราในที่ประชุมนะครับFinally, I would like to say something. On behalf of the Department of Medical Science, I'm extremely grateful to Dr. Arongsi Achiniyaskun for organizing this seminar with us. I would like to express my sincere thanks to Dr. Timothy Stout and Dr. Mark Pennesi for kindly contributing the time to educate our personnel, including those who are interested in the field of gene therapy, both in this meeting room and who participated in online seminar. And thanks to the working group, including all participants. ก็บัดนี้ได้เวลาสมควรแล้วครับกับผมขอเปิดสัมมนาวิชาการวิทยาศาสตร์การแพทย์และขอให้การสัมมนาในครั้งนี้ได้บรรลุวัตถุประสงค์ทุกประการครับ
ต้องขอกราบขอบพระคุณท่านอธิบดีกรมวิทยาศาสตร์การแพทย์ท่านนายแพทย์สุภกิจ ศิริลักษณ์นะคะที่ให้เกียรติมากล่าวเปิดการสัมมนาในวันนี้ค่ะในลำดับถัดไปนะคะจะเป็นการถ่ายภาพร่วมกันค่ะขอกราบเรียนเชิญท่านผู้ทรงคุณวุฒิท่านคณะอาจารย์จากศิริราชท่านวิทยากรและผู้มีเกียรติทุกท่านถ่ายภาพร่วมกันที่ด้านหน้าขอกราบเรียนเชิญค่ะ
I would like to invite our honorable guests in front of here to for the photography sessions.
ค่ะในลำดับถัดไปนะคะจะเป็นการบรรยายในเรื่อง The Ethics of Gene Therapy นะคะโดยดร. J. Timothy Stout ค่ะ
Ladies and gentlemen, please welcome Dr. J. Timothy Stout to the stage.
ค่ะและในโอกาสนี้นะคะขอกราบเรียนเชิญท่านศาสตราจารย์แพทย์หญิงละอองศรีอัชนียสกุลภาควิชาจักษุวิทยาคณะแพทยศาสตร์ศิริราชพยาบาลกล่าวแนะนำท่านวิทยากรค่ะ
สวัสดีค่ะทุกท่านค่ะ Good morning, Good morning, ladies and gentlemen. It's always a pleasure and an honor for me to introduce our first speaker this morning, Professor J. Timothy Stout. Actually, we already have a 2 days meeting before, so this is a third introduction. Dr. Stout is chair of Ophthalmology Department and director of the Cullen Eye Institute at Baylor College of Medicine in Houston, Texas. His research interests include human gene and cell-based therapy for proliferative and inherited ocular disease, retinal disease genotype-phenotype correlation, and ocular disease gene mapping and discovery. On Monday, Dr. Stout has given us very interesting talks on the overview of gene therapy methods and the surgical aspect of gene therapy, which I think is very important not for the clinician but for the researchers as well. And today he is going to discuss a very important topic, which is the ethics of gene therapy. He reminds me to tell everyone that this is a two-way communication. So please be prepared to answer and discuss with our speaker. Ladies and gentlemen, please join me in welcoming Professor J. Timothy Stout.
ครับคุณเกรด Well, thank you so much, Dr. Achniyasakum. It is truly a pleasure to be here. And this is going to be a different talk from the talks that we've had before. I'm not going to provide any data. I'm not going to talk about viral vectors. I'm not going to talk much about patients. What I'm going to do is I'm going to tell 5 stories. And I want your opinion about treatment for these patients in these 5 stories. And again, as Took mentioned, I really don't want this to be a lecture. I want this to be a conversation. I want you to participate. I will ask you to participate. So let me start off here. Can we get my screen? How do we get that? You know that it's going to be an interesting talk when there's only 6 slides, right? I am going to talk about the ethics of gene therapy, and specifically what I'm going to talk about is where are we going with this? What's appropriate for us to treat, and what's not appropriate for us to treat? And again, I'm not telling you any answers. I'm just trying to make you think about different situations. So when I call on you, there's no right or wrong answers. This is opinion. This is what you think. But hopefully it will get you thinking about things that we as citizens or physicians or scientists or voters need to think about. Well, this isn't not advancing, so I'll just do it this way. Okay, I'm going to talk about 5 stories: human adenosine deaminase deficiency, human RPE65 associated blindness. And this is something that we've talked about for the past couple of days, so most people should be familiar with this. We're going to talk about mouse influenza virus. We're going to talk about mouse NCS gene. And you guys may not know what the NCS gene, but we're going to talk about it. And then we're going to talk about C. elegans, which is a small worm. It doesn't cause human disease, but it's used a lot in research. And we're going to talk about C. elegans and what's called the Dower state. And again, audience participation is required. Okay, let me tell you a story about human adenosine deaminase deficiency. Adenosine deaminase is a protein that's expressed throughout the body. It's required for T cells to work normally. If you don't have adenosine deaminase, you have thank you so much. You've got a severe combined immunodeficiency that's lethal, right? At least it was lethal up until about 5 years ago. In this situation, these patients are born without an immune system, and without any kind of treatment, they die within the first 1 or 2 years of life of some overwhelming infection. I don't know if you remember this, but there was a patient, David Vetter, who was born with this, who was kept in a bubble. This is in the hospital, was kept in a bubble. This was in Houston, Texas. And this patient lived to be 12 years of age before he developed a leukemia, which is also common in patients with adenosine deaminase deficiency. And so he died. But this was a child who was essentially raised in a hospital and raised in a bubble. About 15 years ago, people purified adenosine deaminase and made a lot of recombinant adenosine deaminase and put it into and pegylated it, which is something that will allow it to remain in the circulation for a long period of time. And so patients' lives were extended because they could take this pegylated ADA and it would, to some extent, restore their immune system. It was a very, very expensive drug, and the patients needed to take this medicine twice a month. Very expensive, and presumably for their entire life, right? So patients now, their lifespan was changed from 1 to 2 years to living in a bubble for 12 years to now 20 or 30 years. But it was very, very expensive. About 7 or 8 years ago, people took the adenosine deaminase gene, they put it into a viral vector, they got hematopoietic stem cells, they transduced them, they put them back in the patient, and the gene therapy worked. And now there are a number of patients, this is a brother and sister pair who both had adenosine deaminase deficiency, who now are perfectly healthy. Their immune system is restored. So we've gone from a lethal disease to a non-lethal disease with a one-time treatment. Is this a good thing? Raise your hand if you think this treatment is a good idea. Okay, me too. I agree. I think that we've taken a lethal disease and we've treated it, and it's now no longer a lethal disease, and it's well worth what we all do. All right, I'm going to tell you another story. As many of you know, there is an enzyme, an isomerase, that is expressed in one of the layers of the retina called RPE65. And if you don't have this protein, if you don't make RPE65, you have a disease called Leber's congenital amaurosis. And it's a blinding disease. These are children who will present to ophthalmologists like Dr. Pennesi within the first 6 months of life. They have nystagmus. They can't see. Their parents know that there's a problem. And for many, many years, we've had no treatment. This was a very painful situation where we would have to tell parents, I'm sorry, your child is blind. There's nothing that we can do about this. Approximately 15 years ago, we started to do research on figuring out ways that we could transfer the adenosine the RPE65 gene into the appropriate cells. The clinical trial involved doing surgery. We would go inside the eye, we would take out the vitreous, we would inject a virus that contained the RPE65 gene into the subretinal space. That virus would then infect the appropriate cells and RPE65 would be made. This reversed the blindness. In many patients, this was a near curative effect. One of the ways that this was demonstrated is that we have what's called a mobility maze. And in this mobility maze, we were able to demonstrate that patients before treatment could not navigate this maze because they were blind. And then after treatment, they navigated the maze very, very well. And these patients are all remarkably appreciative of the fact that their blindness has been reversed. It's not a total reversal, but their blindness has been improved. So we've got a drug that, as it turns out, is very expensive, that reverses or stops a blinding disease. Good idea, bad idea? Raise your hand if you think this is a good thing. Okay, so the thing to consider here is now we're treating children actually without their consent, right? It's the parents that do the consenting. We're treating children for a disease that won't kill them. We're treating children for a disease that causes a problem. I mean, they're blind, but it's not a lethal disease. So we're all okay with treating children genetically, permanently changing their genetic makeup without their consent. They're too young to consent, to prevent them or to cure them from not being able to see. I'm okay with that. I do this surgery. I wouldn't say all the time, but I have a case next week where we're going to be doing this exact surgery. And I think that's a reasonable thing. So we have all said that gene therapy to cure a lethal disease is a good idea. We've all said that doing gene therapy for a not lethal disease, but for a disease that causes blindness is a good idea. So we're all on board with this, right? Okay, let me tell you the next story. So in the United States, there is in a state called Maine, up in the northeastern part of the country, there is a large complex that is a mouse facility. They have millions and millions and millions of mice, and this is all for research. It's called the Jackson Laboratories in Bar Harbor, Maine. It's just a wonderful resource. They have knockout mice, they have transgenic mice, and they've got blind mice, they have mice that can see. I mean, it's just a great facility. And this is an aerial picture of the Jackson Labs. And right here is Building H. What do we care about Building H? Well, as it turns out, there is a type of influenza that is totally lethal for mice. The influenza is spread in an aerosolized form. The mice get it, and if the mouse gets it, the mouse dies. So in this building, about 8 to 10 years ago, they had a breakout of this influenza, and it killed hundreds of thousands of mice in Building H. Not every mouse. There actually was a strain of mice. There was a family, a pedigree of mice that actually did just fine. They weren't affected at all. They didn't die. They had no respiratory issues. But it was a very, very rare phenomenon for mice to be able to survive this infection. So they did whole genome sequencing on these mice, and they realized that there was a mutation in 2 separate genes that allowed them to be resistant to influenza. So they cloned these genes and they put them into susceptible mice that didn't have these things. They made transgenic mice, and then they exposed them to the virus, and they survived. So this was a scenario whereby they figured out which genes provided immunity to influenza. So they've gone back and they've asked the question, are these genes present in humans? The answer is yes, they are. A number of years ago, we had when I say a number of years ago, over 100 years ago, there was a huge pandemic in the world, the influenza pandemic, that killed 50 million people throughout the world. There was a a town in Texas called Galveston. It's on the coast, where there was a hospital and they treated a lot of these patients. And this was a horrible, horrible disease. They treated a lot of these patients and they stored blood on a lot of these patients. And for whatever reason, I don't know why, they kept the blood. So these investigators went back and looked at some of these bloods from 100 years ago. And there were some patients that survived and some patients that didn't survive. And they asked the question, well, if these mice have a particular genotype that makes them immune, were there humans that had a particular genotype that would make them immune? And the answer was yes. You know, they were able to demonstrate that there were genetic changes that involved these genes and similar genes that made people resistant to this virus 100 years ago. Okay, you're all familiar with COVID. Some people get COVID and die. Some people don't get COVID. So the question is, well, what about in the United States, we have a term called novid, which is people who've been exposed to COVID many, many times and have never really gotten the disease. So the question is, are there genetic changes in those people that allow them to not get COVID? So let's just say that we had a viral vector that would contain genes that would allow us to be immune to influenza or COVID. And we could give these genes through a viral vector to patients to make them resistant to infection by COVID or influenza. Good idea, bad idea? Who thinks that this is a good idea? Raise your hand if you think it's a good idea. Not everybody. So not everybody is saying that this is a great idea. Remember, a lot of us have kind of already done this already. We've already genetically changed our T cells by getting a vaccine. And all of my children, without their permission, got vaccinated for a variety of different diseases, which I don't think that they'll get because they've been vaccinated. But for the people who think that this is a good idea, we're now saying, okay, it's all right to permanently change our genetic code for a disease that we don't even have. It is a disease that we may get, but we don't have it. So it's just an important consideration. So we've all said, boy, you got a genetic disease that's going to kill you. Let's do gene therapy. You got a genetic disease that's going to blind you. Let's do gene therapy. You've got no disease, but you're at risk for disease. And about half of us say, yeah, that's a good idea, right? I think pretty much my guess is, who in this room has been vaccinated for COVID? Raise your hand if you've been vaccinated for COVID. 100% of everybody in this room has been vaccinated for COVID, right? So for the people who say, well, I'm not so sure about gene therapy to prevent influenza, just think about it. I mean, you've already gotten a shot in your arm that permanently changed your T cells so that you wouldn't get COVID. And thank God for it. I mean, nobody wants COVID, right? Okay, let's go to the next story. Okay, let's talk about the mouse NCS gene. As it turns out, you can develop a maze, and this is an image of a maze here, and the mouse comes in here, and you put something over here, a little bit of cheese, something like that, and the mouse will figure out a way to actually get to the cheese. And these mazes can be less complicated or more complicated, but it's a maze. And if I take this mouse and I put this mouse in this maze, it will, and I'm making this up, but you get the idea. It'll take him 10 minutes to get to the cheese. If I then take that mouse and I put another piece of cheese in and I start the mouse over here, it's going to take 9.5 minutes for the mouse to get to the cheese. And if I do it over and over and over and over and over, it'll take seconds. I mean, the mouse just knows exactly how to go to get the cheese. There's a learning curve, and the mouse learns what the path is. If I then take that mouse and I put him back in the colony and I leave him in the colony for 6 months, and then I bring him back to the maze, it takes the mouse 10 minutes to get to the cheese. In other words, the mouse has forgotten what he's learned. If I put him back in the maze, he'll figure it out faster than he did the first time, but it's still a path that he has to figure out, right? It still takes some time. If I take this mouse and I train him to do this, oops, now it's working. If I train this mouse every single day for a month, and then I take the mouse out, put him back in the colony for a month and bring him back, it takes him a very short time, but not just seconds, to figure this out. So clearly there's memory and memory decay in these normal mice. You can see where I'm going with this. So they actually, at Jackson Labs, the same labs, they actually found mice that you could train them how to do this maze, and then you put them back in the colony for 6 months, and then you bring them back, and they remember exactly how to get there. And it takes them seconds to get to the cheese. Another thing, if I take this mouse, a normal mouse, and I put them in a maze and I train them in the maze, and then I change the maze, it takes them 10 minutes to get to the cheese because they don't know the new maze. If I train the mouse on 10 different mazes, pretty soon they start to get confused and can't remember the first maze from the second maze, right? And they lose, I wouldn't say that a mouse has concentration, but they lose the ability to do this. So you can overload them with newer mazes, and they start to forget the earlier ones. These mice at the Jackson Labs, you could put them in maze 1 and they would figure it out. You could take them out for 6 months and bring them back and they knew exactly where it was. You could actually train them on 10 different mazes and take them out, and they would come back and they would remember exactly what the maze was. Some of these mazes became remarkably complicated. So this is a relatively small maze here. They have some mazes that are 10 feet by 10 feet in size, and these animals could learn those difficult mazes. You could bring them back in 6 months and they still remembered exactly how it was. So this brings up the point that there is something about these mice that allows them to learn and or remember a difficult task. So they cloned, they did whole genome sequencing, and they bred mice and they made transgenics, et cetera. And they found that there was a cluster of genes that were controlled by something called NCS1 TG. When they took these genes and they put them into normal wild type mice and made those mice transgenic, they now behaved in a much, much faster way of navigating the maze. It wasn't perfect. It wasn't like the special mice, but they were really pretty good, and they clearly learned a lot better. So what's the next experiment that you would perform? Mark, let me ask you. Okay, so the next experiment would be to knock out this gene cluster. And when they did that, they produced mice that could find the cheese, but they never could learn. They could never get past the 10 minutes. It was always trying to figure it out. The next thing that they did was to ask the question, well, do these genes exist in humans? Guess what? They do. We have these genes. They then asked the question, well, do people with eidetic memories, photographic memories, do people with photographic memories have changes in this gene cluster? And the answer was not really. They really didn't. So we don't really know why people who have photographic memories have photographic memories. But what this tells us is that there's probably a genetic basis to learning and memory. And in a simple system like a mouse, we're beginning to understand exactly what contributes to this. Okay, you can see where I'm going, right? Let's just say that we figure it out. Let's say that we find that that's in humans, and we put it in a viral vector, and we get a viral vector that can infect the brain. Good thing, bad thing. Who thinks that a drug that would improve memory and learning would be a good drug? Raise your hand if you think it's a good idea. Okay, we've got relatively few people thinking that that's a good idea. Here's the problem. If a drug like this is ever produced, you guys are going to rule the world, right? The rest of us are going to be handicapped. I can imagine not treating my children and having most other children treated with this are going to put my children at a disadvantage in the future world, right? So I also am opposed to this because I'm not preventing disease. I'm not curing disease. I'm making our current state better. And that's where I start to get a little uncomfortable. Yeah, gene therapy for a lethal disease, I got it. Gene therapy for a blinding disease, I got it. Gene therapy for a potential disease, I got it. Gene therapy to make me smarter, faster, whatever, I'm a little uncomfortable with that. But there's no right or wrong answer. I just need everyone to think about it because I can assure you that although we don't know what the human analog to this is, we will sometime. And I will also predict that everybody in this room who said, no, I don't want to do that. When somebody else is starting to do that, your opinion may change. When you don't have the memory that those guys in the back there have, you might be in trouble. Okay. One last story, and then I'll quit. So there's a worm called Caenorhabditis elegans, C. elegans, and it's a multicellular worm, and it looks like this. And here's its head, and here's its tail. And we understand a lot about how these worms behave. We understand a lot about how cell determination takes place. There was a scientist in the UK who won a Nobel Prize because what he did was he mapped every single cell in C. elegans from the first cell. So he took one cell, it was pluripotent. Then you had two cells, then you had three cells, then you had a larva, and he mapped every single one of those cells, what they ultimately became, and when the gut was formed, and when the mouth was formed, and when the tail was formed, et cetera. So we really know a lot about these animals. Now, these animals they have a lifespan of 90 days. You're born, you exist for 90 days, you're alive for 90 days, and then you die. And that's true for all C. elegans. That's true for almost all worms. There's not a lot of variation. There's not an animal that dies at 2 weeks and then another animal that dies at a year and a half. It's pretty much all about a 90-day story. But there's something that's really interesting about these animals. And that is that at the larval stage, if you put them under high stress conditions, and by high stress, I mean, you remove nutrients, you take away the sugars, you take away amino acids, you alter the temperature, et cetera. If you put them into high stress, they actually go into a state called the Dower state. And you can remain in the Dower state for as long as 2 or 3 months. And then if you wake them back up by resupplying the nutrients, they'll just go right back to having a normal worm life, whatever a normal worm life is. But the clock doesn't change. So in other words, and you can actually do this to not just larvae, you can actually do this to adults. You can put the adults in a condition where they go into this, I don't want to call it hibernation, but it's going to be something like that. And if you do this to an adult that is 45 days old, and you put the adult in the Dower state, for a month, and then you reverse it and you give them the nutrients back and they come back, guess what? They have 45 days left to live. So there's a clock that remembers how far you are in your life. And even though you might have gone into this dour state, this hibernation kind of state, when you come back, you're still only going to get 90 days. And if you put an adult who's 80 days old into the dour state, and then you bring them back a month later, guess what? They got 10 days. So there's a clock that remembers exactly how far you are in your lifespan. So the entire genome of C. elegans is well understood. And so there are a number of, I won't go into how they did this, but there was a number of very, very clever experiments that looked at the genes that allowed you to go into the dour state and the genes that allowed you to come out of the dour state. And they started to play around with these genes, and they did a series of experiments where they did random mutagenesis of these genes. And guess what? They produced, you can make a transgenic C. elegans, not a problem. They produced a C. elegans line that instead of living 90 days, lived 200 days. Same kind of thing, right? You could be at 100 days in, a lot old, at least 10 days older than a normal C. elegans. And you could put them into the dour state, you can bring them back out of the dour state, and they would have another 100 days to live. So what these guys have shown is that there are genes in C. elegans that are responsible for lifespan. So they went back and they said, do humans have the same genes? And yeah, we got some like it. It's, not exactly the same, but we've got some like it. And then they went to people who were 100 years old and compared them with people who had passed away at 50, 60 years of age. And they asked the question, are there changes in these genes that are associated with this long life? They actually had one person who was 123 years old. And the answer was no, there didn't really seem to be a lot of changes in those genes. So we don't know what affects our lifespan. I don't. Right now, I don't know. I know there's a bunch of bad things that I can do that will shorten my lifespan. Driving a motorcycle on Sukhumvit Road is probably not very good for my lifespan. But there are a lot of things that you can do that can shorten your lifespan, but that ultimately our lifespans are controlled. Our life expectancy is different from our lifespan. Our lifespans are probably controlled by genetics. So it's very interesting to me that there is clearly a eukaryotic organism whose genes I understand and whose genes I can manipulate to change their lifespan. So guess what? I can't tell you exactly what's going on with humans, but I'm pretty sure that our genes influence the fact that we can live to about 100, 110 years of age. And after that, we're not going to live any further. So here's a question. Let's just say that I figure out what those genes are. And let's just say that I put them into a viral vector. And let's just say that I can infect all the cells in your body. Good thing, bad thing? Who thinks that's a good thing? Raise your hand. Nobody thinks it's a good thing. I'll ask you the same question when you are 80 and you are, thinking about, well, gosh, the drive to live is pretty high. So as much as all of us would kind of say, oh no, this is a really bad idea, this would be a very profitable drug, right? This would be a drug that would make zillions of dollars. But it's clearly something that we need to think about. It's clearly something, the problem is when these papers first came out, there was a lot of discussion about whether or not altering human lifespan was a good idea. And there was a very, very interesting paper that said, if you change human lifespan by 5%, so in other words, instead, let's just say our lifespan's 100 years. If you change our lifespan by just 5% and pushed everybody up to 105 years, we don't have enough water. So that would really pretty much be the end of our planet because we don't have enough water to sustain that. But be that as it may. So nobody here thinks that extending life right now in this room when you're 30 years of age thinks it's a great idea. We'll see what you think when you're 80. But again, the reason I tell these stories is because we need to start thinking about this stuff. We need to start thinking about what gene therapy does. So as molecular biology defines the human condition, what should we treat? Everybody here said lethal disease. Yeah. Everybody in here said blinding disease. Yeah. Some people in here said infectious disease possibility. Okay. Nobody said memory and nobody said lifespan. But these puzzles will be solved. I mean, I can't tell you exactly when, but certainly within, I'd say 5 to 10 years. And so we need to start thinking about what we're going to do when we understand this stuff. And with that, I will end. My lifespan has come to a close. So thank you all very, very much for inviting me. It's been wonderful. I'd be happy to answer any questions. Yeah, it's really not kind of one of those things where you've got a lot of questions. This is one of those things where you just gotta think, wow, that could be interesting. Anyway, so thank you all very much. I appreciate it.
เดี๋ยวเราจะพักเบรกกันเป็นเวลาประมาณ10 นาทีนะคะสำหรับท่านที่เข้าร่วมในห้องประชุมสามารถหยิบอาหารเด็กได้ที่ด้านข้างห้องได้เลยนะคะ
For the next, we would like to take a break for 10 minutes, and we'll come back at session 2 around 11:25.
นะคะเราจะเข้าสู่ช่วงที่2ค่ะซึ่งจะเป็นการบรรยายในเรื่อง Information and Gene Therapy nanoparticle and CRISPR knockout monkey ค่ะโดยท่านวิทยากรของเราในช่วงที่2นะคะจะเป็นดร. Mark Pennesi นะคะโดยท่านเป็น Professor in Ophthalmology ที่ Oregon Health and Science University ค่ะ
So for the next session will be the talk about the inflammation and gene therapy, nanoparticle and CRISPR knockout monkey by Dr. Mark Pennesi, the professor of in ophthalmology at the Oregon Health and Science University. But for now, I would like to invite Professor Dr. Laongsi Achnia is going to give us an introduction speech before the guests give the talk.
Ladies and gentlemen, our next speaker is Professor Mark Pennesi. Dr. Pennesi is a professor of ophthalmology at Oregon Health and Science University and chief of the Ophthalmic Genetics Division at the KCI Institute. He's interested in retinal diseases, and his research focuses on developing novel treatments for inherited retinal diseases. Most notably, he led the first ever CRISPR gene editing procedure to restore vision in partially blind individuals. On Tuesday, Dr. Pennesi has explained to us the outcome measures used in ocular gene therapy trials with respect to the FDA guideline. He also talked about the X-linked retinoschisis trial, Luxturna, X-linked retinitis pigmentosa, and the Editas gene editing trial. And today, Dr. Pennesi will talk about the inflammation and gene therapy, lipid nanoparticle, and CRISPR knockout monkey. Ladies and gentlemen, please join me in welcoming Professor Mark Pennesi.
Okay. All right. Well, the good news is for my talk, audience participation is not mandatory, but you're welcome to ask questions and participate. I'd like to start off by just saying thank you. It's really a big honor to be invited here. This is my first time to Thailand, and I really am loving it. And I'm really enjoying meeting new colleagues, and hopefully we can form new collaborations. I think there's a lot that we can do together. There's a lot of potential for treating inherited retinal degenerations here. On Tuesday, I talked a little bit about the clinical trials and some of the gene therapy that we have done. Today's talk is going to be a little different. I'll talk a little bit about some of the problems that we've seen with gene therapy. And I think this is important to really look at, in particular, inflammation with gene therapy. And even if you're looking at other diseases, I know that I've learned that thalassemia is very common here, and potentially you could do gene therapy for that. But inflammation might be a problem with any kind of gene therapy. And I think we can learn a lot about inflammation from gene therapy from the eye. And it turns out the eye is a particularly good place to learn about inflammation, because we can actually look into the eye and see the inflammation in real time, and I'll show you that. And I know that many of you are not ophthalmologists, so I will sort of explain to you and walk you through some of the terms that are specific to the eye. In the second part of the talk, I'm going to actually go a little bit into basic science and talk about some of the new therapies that we're working on, as well as animal models. This is all very new data. So it's still very much a work in progress, but I'm excited to share it with you. So before I go on, I'd like to acknowledge my colleague at KCI Institute, Dr. Paul Yang, who is a uveitis specialist, that's inflammation in the eye. And he's really been the one who has helped me learn a lot about this topic. And inflammation in the eye is actually pretty complicated. We see diseases that cause inflammation even without gene therapy, such that there's a whole specialty called uveitis. And sometimes that can be due to infections, but many times there's not an explanation as to why patients get inflammation in the eye. There are certain HLA factors that may predispose you, but we really don't fully understand it. So one thing, though, that we've learned or realized is when we started doing gene therapy in patients, we've started to see something a little bit new and something that's a little bit different than the inflammation that we've seen in the past. And we've come to call this gene therapy associated uveitis. And what is the definition of that? Well, we say it's really any inflammation within the eye that is greater than what we would expect from the surgery itself for the gene therapy. And we can sort of define that in different ways by how severe it is and by where it is in the eye. And there are a number of standardized criteria that will say that it's in the anterior part of the eye, the anterior chamber, or within the gel of the eye. That would be an intermediate uveitis, or in the back of the eye in the retina. And so you can see here, there are these little white dots on the surface of the cornea. And these are actually cells. So these are actually inflammatory cells that we can see, T and B cells. So this is what makes the eye really useful for studying inflammation, because I can just look at the patient and I can actually measure the information. I don't have to take a blood test or do a biopsy. I can visualize the inflammation. And if I look in the gel of the eye, I can also see these cells. And sometimes underneath the retina, we can also see inflammatory cells. We can also define it as if it's just a single episode, or if it's recurrent or chronic, if it comes back. And inflammation sometimes can be very difficult to treat, because you'll treat it with steroids, it'll go away, you'll stop the steroids, it'll come back, and you have to keep doing this. So this is sort of still very much an emerging concept, and we're learning a lot. And I'm going to talk about a little bit one of the trials I think that really started to teach us about this. And I talked about this gene therapy trial on Tuesday, and this was a gene therapy trial for a disease called X-linked retinoschisis. And this is a disease that occurs only in men. The gene is called the RS1 gene. And you can think that this gene is a little bit like glue. It kind of holds the retina together. And so when you're missing the gene, the retina actually splits apart, and you can see these cavities here that form. And this decreases the vision of the patient. They can get retinal detachments. And so it can be a disease that causes a lot of vision loss. As I described before, we've developed an AAV2 therapy to put a copy of this gene back into the cells. And this is delivered intravitreally into the vitreous. So this was the design of the clinical trial. And like all clinical trials, we typically do a dose escalation. So we started with a very relatively low dose of 1 × 10^11 vector genomes per eye. And then we increased to 3 × 10^11 and eventually to 6 × 10^11. So what we did observe was inflammation in many of these patients. And this was a trial in which we were really learning in real time, because we didn't know what was going to happen. But we did see quite a bit of inflammation early on. And I'm going to kind of show you a little bit about what that looks like. So one of the common things we saw in these patients is something called stellate chorotic precipitates. And that's these little white dots on the inside of the cornea here. And you can visualize these in the patient when you shine light into the eye. And these are, typically conglomerations of white blood cells. You can often see these in the context of a viral infection in patients who have, sometimes rubella or other types of viral infections. So it's kind of interesting that we're putting a gene therapy that's based on an AAV vector, and we see a response that looks somewhat similar to what we see with viral infections. Now, these aren't necessarily harmful, but if you have inflammation for a long time, that's not a good thing, and that can cause problems. So it's a sign that we need to treat the underlying inflammation. So now, if we look back into the gel of the eye, we can also see similar cells here. You can see these little white spots, and that's called a vitritis, meaning inflammation of the vitreous or the gel of the eye. And if we do an ultrasound, we can see that as well. So here's the data from our trial. And what I've done is I've graphed out the inflammation in the first 6 patients that we treated. And the 2 different colors represent whether the inflammation was in the front part of the eye or in the back part of the eye. But you can see that in 4 out of the 6 patients, we did see these signs of inflammation, these little cells. And so we don't leave this alone. What we did was we actually treated these patients first with topical steroids, which in many cases that was enough to make the inflammation go away. But in a few patients, we had to give them oral steroids as well. The good news is in all of these patients, the inflammation disappears with treatment. However, now when we go up to a little bit higher dose, now we're starting to see more inflammation, and we're having to treat the patients with higher doses. As we went up, then most of these patients really needed oral steroids to take care of the inflammation. And so at this point we said, maybe we should pre-treat the patients even before we do the gene therapy. We should give them steroids first, make sure we immunosuppress and see if that helps. So we did that, and still at the highest dose, we saw inflammation. Once again, with enough steroid treatment, we were able to get this inflammation to go away. But you can't keep patients on oral steroids forever. So it's important to be able to eventually get them off the steroids. Now, one thing that's kind of interesting is when you treat a patient with AAV, you develop neutralizing antibodies to the AAV vector. And that's shown here in red. And what you can see is that almost every patient had an increase in antibody, these neutralizing antibodies. However, it didn't necessarily correlate to which patients got inflammation and which didn't, which was kind of interesting. The other thing was we actually pre-screened many of these patients before we treated them to make sure they didn't have a high baseline level, because many of us get exposed to wild type AAV and we may have antibodies. So this just shows the rest of the patients. And in total, we ended up treating 27 patients in this trial, including children. And as I mentioned on Tuesday, unfortunately, the gene therapy really didn't seem to work. But we did learn a lot about how inflammation works in the eye. And then, once again, it was kind of interesting because there really wasn't a big correlation between the dose and the neutralizing antibodies. Everyone got them, but it wasn't a clear relationship with how much vector we put in. There also wasn't a really great correlation between the neutralizing antibodies and the clinical degree of inflammation. So that, kind of tells us, well, maybe neutralizing antibodies aren't really the most important thing. There may be other mechanisms such as T cells or innate immunity that we need to think about. Now, unfortunately, there were a few patients who we treated them, the inflammation went away, and then maybe 4 to 6 months later, the inflammation came back. And so this would be what we call recurrent or chronic uveitis. And so 3 out of the 27 patients did develop this chronic uveitis. And that's a problem because we can't keep patients on steroids forever. Steroids will cause cataracts or clouding of the lens. It causes, weight gain, all kinds of high blood pressure. So eventually we had to get the patients off the steroids. And so we ended up starting these patients on other forms of immunosuppression, specifically methotrexate, which is a steroid-sparing immunosuppression. And this just shows one of the patients who had all of these little cells in their eye. And if we plot them out over time, the red shows when we put them on methotrexate and the inflammation got better. However, a lot of times patients don't like to stay on these drugs even for a long time because you have to get your blood checked. And during COVID, follow-up was very hard. So eventually the patient got off of the methotrexate at the pandemic. Fortunately, he stayed in remission and the inflammation went away. So this was a success. This was a second patient who showed a very similar pattern, eventually had to go on methotrexate. And unfortunately, because of the pandemic, he was unwilling to come back in. He was from another state. And we said, we can't just keep prescribing you these drugs if you're not going to come in. And so we lost him to follow up, unfortunately. And then the final patient, similar story, the methotrexate helped with the inflammation. Eventually, though, the patient wanted to go off and the inflammation came back. And at this point, he actually developed chronic changes in the gel of the eye such that it was blocking the vision. And we could go in and take that out surgically, but we decided against that. But this does show that gene therapy can certainly have risk. And I think that it's important that we really think about the immunosuppression protocols. So that was for intravitreal gene therapy, where we put the virus into the gel of the eye, but we often put the virus underneath the retina. And I'm going to show an example of another patient who developed inflammation from when we put the virus underneath the retina. Now, the good news is that the space underneath the retina tends to be immunoprivileged. And so I think that the risk of inflammation is less. And we see a lot less of these kinds of cases than when we put the virus into the gel of the eye. But this can happen, and we need to be honest about it and realistic about it. So this patient got a gene therapy, and it was completely uneventful. The surgery went very well. But a week after the surgery, if we look at the retina, what we can see is that there's fluid underneath the retina. And normally, when you inject fluid under the retina, it goes away in about 24 hours. So there really should not still be fluid underneath of the retina. And if you look at this scan of the retina, you can kind of see these bright little spots, and you can also see them here in cross-section. And we don't know what these are because we can't sample them, but we think they're inflammatory cells, or at least that's one hypothesis, that these are inflammatory cells. Now, over time in this patient, we started to see chronic changes in the eye that was treated as compared to the other eye. So that fluid went away eventually, but there still were these kind of bright deposits underneath of the retina. And we treated this patient with pretty high dose steroids, immunosuppression, and really didn't see a big difference. But you can kind of see over time, there are these dark spots developing in the retina. And now about 4 years later, you can see there's sort of this discolored area, and that actually correlates to where the gene therapy was delivered. The retina has thinned in that part, and the vision is worse than when the patient started. So this is very humbling. You know, we set out to do something good. We set out to try to restore vision. preserve vision. But we have to admit that sometimes things go wrong in trials, and there is risk to these trials, and patients can lose vision. And these kinds of cases you always feel very bad about because you have to keep seeing the patient. But we need to really think about this, and we need to be very careful going forward and be very cautious and realize that what we're doing is new, and we still don't fully understand the implications of putting gene therapy into the retina. Now, I don't want that to scare you. I think that this is something we still need to do, but we do need to be cautious. And, this is something we talk about a lot when we're thinking about a new gene therapy, and how much immunosuppression should we give? Should we give it before the treatment? And then even after the treatment, how long should we keep the patient on immunosuppressant? Should it be a month? Should it be 2 months? Should we use prednisone? Should we be using other drugs? And these are still, I think, big unanswered questions, and really a topic of debate. There's sort of benefits of treating very heavily, but then there are also disadvantages because many of these drugs have their own side effects. Okay, so I'm going to shift a little now, and we're going to talk a little bit more about basic science and some of the newer things that are coming along for gene therapy. And maybe this is a way to get around some of the problems that we have seen with viruses. And so the next part I'm going to talk about is actually trying to deliver gene therapy with lipid nanoparticles. And the person who's really been responsible for this work is Renee Rouse at our university, and I want to give her full credit. This is her work. This is still very much a work in progress. But as you've heard in the previous days, lipid nanoparticles are complex structures that are composed of different phospholipids, cholesterol, different pegylated lipids, but essentially you can package RNA or DNA inside of them. And by modifying different moieties, we can target them to different cells. Lipid nanoparticles have been FDA approved for a variety of therapies. They can be safe, they're scalable, there's a lot of different modules that you can add to them, and we can have multiple different cargoes. Finally, I think what's really important is we can deliver larger genes. So with AAV, you're limited to about 4.5 kilobases. With lipid nanoparticles, we can definitely go larger. I'm not sure what the upper limit is, but I think they have the potential for delivering bigger genes, which in particular, something like Stargardt's, which is a common blinding disease in children, that's going to require a larger delivery method. Okay, so this is some of the preliminary basic science work looking at these lipid nanoparticles. And what you're seeing here are these green HAP1 reporter cells. And these cells express EGFP, and then they have a stop codon in front of an M-cherry gene. So that normally does not get expressed. But if we add a prime editor to these cell cultures, a prime editor that's enclosed in one of our lipid nanoparticles, it will cut out that stop sequence, and you can see that these genes will turn, or these cells will turn yellow. So this is kind of the first step to show that we can get lipid nanoparticles to do gene editing in a culture model. So the next step was to try to actually go to iPS cells, so human-derived iPS cells that have then been differentiated into retinal pigment epithelial cells, and taking these lipid nanoparticles and engineering them to express GFP. And so you can see, indeed, we're able to get the lipid nanoparticles to transfect these cells and express GFP. You can see that there is kind of a dose relationship here, which, too much, actually, you end up getting less expression, which is interesting. But so this is the second stage, right? Now we're into human iPS cells. So we wanted to really see if we could take this up to the next level. And so we went to non-human primates, and our surgeon, Dr. Andy Lauer, was able to take these lipid nanoparticles that were packaged to express GFP and inject them underneath the retina in the primate. And so here's the procedure shown here, and you can see this bleb that is formed as the fluid is injected, and you can see how it's already glowing. That's because they put a little GFP in the initial solution, but that dissipates. So he made two areas where he put in these lipid nanoparticles. And so 48 hours after injection, you can see that there is fluorescence. And we can see that a little bit better shown here. So these two different blebs. And later on, if we do histology, we can see where that expression is coming from. And it primarily looks like it's in the retinal pigment epithelium. And that's not too surprising, because the retinal pigment epithelium is a cell that likes to eat things and phagocytize things. So it's fairly easy for those lipid nanoparticles to get taken up by the RPE. And so as a first level, that's a good thing, but we really want to get into other cells. And this just shows you, if we do co-staining with a marker for the retinal pigment epithelium, it really does look like they're there. So next, though, we were able to modify the lipids to try to target other cells. And we have been able now to get these into the photoreceptors. And so if you see here, these are actually cone cells and rod cells. This is a rod marker in red. And so we see some cells that are yellow, meaning that we've targeted some of these photoreceptors. Now, this is still not a super efficient process, but it's something that I think now it's become more of an engineering problem. This just shows a little bit zoomed out picture. We've also been able to make other tweaks to the lipid nanoparticles to target Mueller cells in the retina. Unfortunately, we've not been able to get bipolar cells to target yet. So what about an immune response? I think one of the big questions is, well, are these lipid nanoparticles less inflammatory than AAV, right? That would sort of be our hope. And if we look at, remember, we made two blebs, and if we look at the first bleb, we really don't see much inflammation. This is a marker for IBA1, which looks at microglial cells, also at CD3, and really not a whole lot of change. However, in the inferior bleb, we see quite a bit of staining of IBA1. We also see this material beneath the retina, which is probably inflammatory cells. So there is some inflammation here. When we look at the histology of the first bleb, really don't see maybe some mild changes. But in the second bleb, definitely some inflammation and thickening of the choroid. So I think that although this is still very early in a first attempt, we have to say, okay, it's possible we can get inflammation with lipid nanoparticles. And we need to understand that. Now, a few things, this is still very early. These animals were not put on any immunosuppression ahead of time because we purposely wanted to see if they would develop inflammation. We really still need to characterize this. So in conclusion, we can get these lipid nanoparticles into RPE, photoreceptors, and Mueller glia, but we are seeing some inflammation. This type of experiment's been repeated in the mouse now, but there's still a lot of work to be done, I think, before we can put these into people. So the final part of the talk is I'm gonna shift over to another little bit different topic, but also I think a very exciting topic, which is looking at new models for retinal degeneration. And as we heard on Tuesday, many genetic diseases can be modeled in mice, but it turns out that mice are not great for a lot of eye diseases because they don't have a macula, which is the central part of the retina. And that's the part that we really care about. That's the part of your vision that gives you high resolution and lets you read. So can we make animal models in primates? And this has been really the work of Martha Nuringer, who has been at our primate center, the Oregon National Primate Center now for decades. And she really is the person, I think, who has discovered and characterized these animal models. And I think what she's done is really exciting, and I'm really excited to show you some of this work. So out at the Primate Center, there are three different ways to get models. So one is to just look at naturally occurring models. The Oregon Primate Center is one of the largest primate centers. They have over 2,000 primates. Many of them, these colonies have been there for years. They're interrelated, their families, they've mapped out a lot of the genetics. And sometimes just by chance, they'll observe a problem. The second way is to actually do whole genome sequencing and to look for carrier mutations that you might be able to mate and create an animal model. And the third way is to use gene editing to actually introduce a novel mutation to develop an animal model. So there are a number of naturally occurring models at the Oregon National Primate Center. One is something for drusen, and drusen are these little yellow spots that you see in the retina. These are characteristic of age-related macular degeneration, which is one of the most common forms of vision loss in the elderly. Many of us will have had older relatives with macular degeneration. So interestingly, as these animals get older, they also develop drusen. And if you put them on a Western diet, like of McDonald's, they actually get more drusen than if you put them on a healthy diet. So this is something that we can use to study. Interestingly, though, these animals don't get thinning of the retina like humans do. So there's some limitations to this model. There's a second model, which is actually genetic, where in the Japanese macaques, they also develop drusen, but this is dominantly inherited. So there is a genetic component. And we've looked very hard for the gene, even doing whole genome sequencing, and still haven't really found it. So that's still a little bit of a mystery. There's another naturally occurring model in the monkeys, and this is for a really terrible disease. This is something called neuronal ceroid lipofuscinosis, or Batten's disease. This is actually a disease that Tim and I see in clinic, and it's a very sad disease because oftentimes a young child will come in. They'll often have had rapid vision loss over the course of a year. And you look in at the retina and you see very severe thinning. And then a year or two later, they start showing neurological signs and they start having seizures, and ultimately many of them die. So this is a really devastating disease. They've actually now found a few of the animals, many of the animals actually are carriers for one of the genes, CLN7, and they found a couple of animals who actually have this disease and show the retinal degeneration. They also get neuronal degeneration, and they're very sick. So this is one we're actually trying to do gene therapy on to see if we can slow down the disease. Bardet-Biedl syndrome is a rare genetic syndrome. You've probably not heard of it if you're not an ophthalmologist. But in this disease, the patients have many interesting symptoms. They get a problem with their retina. They tend to be obese. It's very common for them to have extra fingers or toes. and they get problems with their kidneys. And we've also found a model of this in the monkeys. And so far, they found 3 cases. And when we look at the function of the retina in these monkeys, you can see that they get thinning of the retina. And if we do electrophysiology, it's very abnormal. So they don't see well. This is something also that we're looking to do gene therapy for. Now, once we find these naturally occurring mutations, you can try to make more animals by doing in vitro fertilization. So this was the first attempt at that, and it was successful. And this is the monkey here. And what you can see over time with the retina is it's getting brighter. And what that means is that it's actually getting sicker over time. And if we look at the thickness of the retina, it's thinning, and the electrophysiology shows the decline, and also the visual acuity is lower. So we definitely have a model now of this particular disease, and we can work on gene therapy. This disease is not modeled very well in mice. So this will be something I think that will really help us get to a human treatment quicker. So I mentioned before trying to find these pathogenic variants in the population, and they've done a large screen at the Oregon Primate Center. And interestingly, they found a number of monkeys with mutations in EYS, which is a gene that causes retinitis pigmentosa. And it turns out this gene is actually, I think, the most common gene in Thailand. So it's possible that we could eventually breed these monkeys to make a primate model of this particular gene. And a number of people here are actually working on gene editing for EYS. So I think this is very exciting because we may be able to develop a primate model that you could work on and use your gene editing on. We've also found animals with genes in albinism, and interestingly, these animals have lighter color pigment. So we'll be developing that. The final part I'm going to talk about, I think, is sort of the most exciting, because there are, once again, genes that aren't really good in mice, or the models do not really recapitulate the human disease. And one of these is a disease called Usher syndrome. This is a really difficult disease because the children are born deaf first. And then usually, maybe the age of 5 to 7, they start to lose vision. So not only do they have hearing loss, which can often be treated now with a cochlear implant, but then they also go blind. If you knock out the most common gene, myosin 7A, in the mouse, you really don't see a phenotype. And so the mouse model is not very useful. People have been working on gene therapy for this, but we don't really have a model to test it on. And one of the things that has been realized is that in the primates and humans have structures that are not in the mouse. And this is the photoreceptor. And there are these little fingers on the photoreceptor called the calyceal processes that are not present in the mouse. And we think this might be one of the reasons why when you knock this gene out, you don't get a disease in the mouse. So the question was asked, well, can we use CRISPR to knock this gene out in the monkeys? And we have a very, very talented scientist at the Oregon National Primate Center named John Henbold, who has worked on this. And he said, I think I can do this. I think I can knock out myosin 7A using CRISPR. And so this is done at the embryonic stage. So shortly after fertilization, he injected the Cas9 with guide RNAs to target the myosin 7A gene. We then let this proceed to the blastocyst stage. You can take a biopsy and do sequencing. And the first time we did it, we got about 50% knockout. And why was that? Well, it must have split before the CRISPR did the edit. And so we ended up with a mosaic monkey that actually didn't show any retinal degeneration. The second time, or the second attempt, was 100% knockout. And that actually led to a live birth. And this monkey was named Gemma. And Gemma is the first full knockout of myosin 7A. They were able to measure the hearing early on and confirmed deafness was present at 1 to 2 months. Also, there's vestibular dysfunction, so abnormal balance, kind of a wide gait. And we're starting to see now a change in the photoreceptors or a thinning of the photoreceptors. If we measure an electroretinogram, which is a electrical measure of the retinal function, we see a decrease. So indeed, this does recapitulate all of the features of Usher syndrome that we see in the human. So, this has been an incredible amount of work. As you can see, many people were involved. I think this is really very promising because one of our colleagues now has developed gene therapy for myosin 7A, and we're going to actually try to treat these animals. And hopefully that will lead to human trials. So in conclusion, you can see there are some barriers to gene therapy with AAV in terms of inflammation. I think there's still a lot we need to understand, but there's still a lot of hope in the future. We have all of these new technologies coming along that are also very promising, and new technologies to model these diseases better and try out these new therapies. As I talked about on Tuesday, we've also used CRISPR now in humans and shown some benefit. So I think we're going to see changes, very soon. We'll be able to treat rare diseases. This is still very expensive. We need to figure out better ways of scaling it, making it more affordable so that all of these patients can be treated. And I really look forward to working with you and collaborating with you to do this and help patients. Thank you very much.
ในวันนี้นะคะต้องขอกราบขอบพระคุณนะคะท่านวิทยากรของเราทั้งสองท่านค่ะท่านดร. เจ ทิโมธี สตาวและท่านดร. มาร์ค เพนเนซีที่มาบรรยายให้กับพวกเราในเรื่องจีนเทอราปีค่ะในโอกาสนี้ถ้าท่านใดมีคำถามสามารถยกมือได้เลยนะคะ
Thank you very much for very nice presentation. I learned a lot. So actually, I learned all of your presentation. I learned it first time here. And I have the question about lipid nanoparticle. And we know well about the lipid nanoparticle in Pfizer and Moderna vaccine. And the cost of each vaccine is just like $10 or something. And I'm just wondering if the lipid nanoparticle for gene therapy is going to be a lot cheaper than the adenovirus or some form of gene therapy.
Yeah.
Do you have some idea about that? Thank you.
Yeah, I think you asked the $1 million question, literally, because that is about the cost of the first gene therapy, Luxturna. It's about $800,000. And that's not really practical in the long run. We need to figure out a way to lower that cost. And I think that probably will happen with time. If you think about cell phones, the first cell phone was like a suitcase that you carried around and, only the richest person, could afford it. Now we all have cell phones. So I think the cost of gene therapy will go down. But I still think that that's not enough, because when we think about all of the rare diseases, even within the retina, there are probably 300 different genes. And so the cost of developing gene therapy for all of those is probably not feasible. It might be feasible for some of the more common diseases, but there are some genes that are so rare that maybe 1 in a million people or 1 in 10 million people have them. And that's really, I don't think it's going to be economically viable. So we do need to find other ways of having gene independent therapies. Now that might be something like neuroprotection, where instead of replacing the gene, the specific gene, you express a gene that prevents cell death or slows down cell death, and people are looking at that. We're going to have other methods, but we do need to find more gene independent ways so that we can really do this around the world and make it affordable.
Thank you very much.
There's somebody in the back there.
Hello, I'm Dr. Kop Gao. Thank you very much for the night talk today. So I have a question. Like, once you inject the viral vector or LNP gene therapy, does that affect the microcirculation of the eye or cause any like microcirculatory changes in the eye?
I'm sorry, you said
microcirculation in the eye.
I'm not sure I understand.
Like cause the
the circulation, microcirculation.
Yep.
That's a good question. We haven't really looked at that in a whole lot of detail. We did do what's called OCTA, or there's a technology called optical coherence tomography angiography, and we measured that in some of our Luxturna patients to see if the blood flow might actually improve, because one of the ideas was, well, if the cells are working better, maybe you'll get an increase in blood flow. I didn't really see an effect on that, but it's also very hard sometimes to image these patients because they have poor vision and they don't fixate, very well. So I think the answer is we don't know.
Is it possible to do that in animal models?
Yeah, certainly. I mean, I think that, with the animal models, we haven't really seen a change in the blood vessels, per se. I mean, at least to my knowledge, unless you have inflammation. But, just the creation of a subretinal bleb, I don't think that changes the microcirculation. Tim, have you?
So we did the same thing that Mark was talking about, where we've done an OCTA on a patient who had Luxturna, and we did it on post-op week 1. So this is 7 days. So you create this blister, and the blister goes away. And it turns out you have air inside the eye, and you have to wait a couple of days for the air to go away. But then you examine the microcirculation with OCTA, and there were changes in the area where the bleb was that reversed themselves by about a month of age, or about a month post-op. So I think the mechanical stresses of putting fluid into the subretinal space can affect the circulation, probably not in a pathologic way. And that's all reversible within a month.
Thank you. I'm Vipal B from Institute of Biological Products, and very nice talk. And I'm very excited for your presentation. So for my question is about the inflammatory, and the first topic. Do you say from the result, it seems like it's not related. The inflammatory is not related from the vector, right? Because when you detect the neutralization antibody, it's still high, but it's not like a case by about the severity of inflammatory. Did you look at the another immune cell like a DC or monocytes on the eyes after inject by the
Yeah, so I can talk a little bit about that. At least I think in that particular trial, we don't necessarily think the inflammation was related to the capsid, but it could have been related to the DNA material within the capsid. And the company that was responsible for that trial did an interesting series of experiments where they tried to use empty capsids versus full capsids and to figure out, what was the source of the inflammation. And it was a little complex. There wasn't a clear-cut answer, but I think there was definitely a component more of the DNA possibly being that. Now, that being said, I think the capsid still can cause inflammation. So, really any component has the potential, the ITR, the promoter, depending on if you have a lot of CPG, motives. So really the other thing is just impurities, in the manufacturing. So, company to company, the quality of manufacturing, may not be exactly the same. So the full to empty, capsid ratio. So, I think there are a lot of variables. It's hard to say that it's just one particular thing.
And about the lipid nanoparticle, it seems like you will put mRNA inside. It's like a Pfizer Moderna vaccine, right? So how do you target exactly on the eyes? And it may cause about inflammatory again, because mRNA in the lipid nanoparticle itself, if it's free in the cells, it can cause of secreted some inflammatory things. So it's made another story of this lipid nanoparticle in RNA carrier.
So with the lipid nanoparticles, I can't go into the details of how they get targeted to different cells because those are sort of proprietary modifications, and that's something hopefully in the future we can talk about, in more detail. But in terms of the inflammation, we do think that the contents probably can cause inflammation because if you try to deliver DNA versus RNA, you get more inflammation with DNA. So, I think it gets back to the idea that there are still, inflammatory components, even if the lipid itself may not be inflammatory, what you put inside potentially can be.
Related to the question about microcirculation, I'd like to ask if you can comment, both of you, about the difference between gene therapy and the cell therapy. Because in the past, there was a paper that they used the mesenchymal stem cells in animal model, and they found the change in the vasculature, like increase the blood vessels in the animal with the cell therapy.
Yeah, I think, are you referring to the Freelander paper with the stem cells where they showed growth of new blood vessels? Or, yeah, I mean, I think there's sort of, I mean, that's kind of a complex question, right? If you put a pluripotent stem cell, it does have the potential to differentiate. I mean, I think ultimately, though, we don't want to do that in the context of inherited retinal degeneration because we probably want to put in differentiated cells or, maybe photoreceptor precursors. The question, really is, how are these stem cells going to work? Are they going to work because you put them in and they release, exosomes or, factors that just help keep the photoreceptors alive? And that might be the case of, J-site, where they're putting these stem cells into the vitreous and, report that they're getting some preservation, versus doing like true regeneration, such as what David Gamm is trying to do, where they're growing, almost like a plate of photoreceptors, and they're going to try to graft them in and get to connect. So I think those are two very different, approaches, and one is much easier than the other. But, the first really will just be protective. The second, I think, would actually be, restorative or regenerative. Tim?
So the short answer to your question is I don't know. But remember, we're talking about two different vasculatures, right? So when we were talking about the OCTA, we were actually looking at the retinal vasculature. So that's the vessels that run in the retina itself. And those blood vessels really only oxygenate the inner 1/3 of the retina. Most of the retina is oxygenated by a blood vessel complex that's beneath the retina, that's beneath the retinal pigment epithelium, called the choroid. We don't really have great ways of measuring choroidal blood flow. At least it's complicated to do, and I've certainly not done it in humans that have had a gene therapy. But in a number of patients who've had human gene therapy, when you look years afterwards, there doesn't appear to be, just on photographs, not a fluorescein angiogram, not an OCTA, there doesn't appear to be any kind of change in their retinal vasculature. That doesn't mean that there's not a change in their choroidal vasculature, but we haven't seen any sign of that.
Yeah, I agree. We haven't quantitated it, but just, I haven't noticed any.
I have another question. Sorry. But you talk a little bit about organoid and in the monkeys model, and I read the news that US FDA just launched the new regulation that we can skip the animal models if we have a good like an organoid model for some of these new therapies.
Yeah.
What is your idea or opinion on that? Can we skip mouse model for this gene therapy so that things can go to the patient quicker? Or is that a bad idea for this kind of new things?
So yes, it's a really good question, and we think organoids are a very useful model. We're putting a lot of resources now towards growing organoids from patients for whom we've collected with different mutations. You can also make them artificially. There are a number of genes, as I mentioned, that are not good mouse models, and you have to use organoids because there just isn't an animal model. And so that was the case actually for the CRISPR trial that we did. The mouse knockout is available. It's an okay model. There's no large animal, model. And they actually did look at the gene editing in the organoids, and the FDA was happy with that. I think that if there is a mouse model available, the FDA would probably still like to see data, from that. What I don't think you need is a large animal model anymore. You'll still need to do safety though, likely in a non-human, primate. So I think organoids can be very useful for showing some kind of proof of biological efficacy, but they're not going to be sufficient for showing safety.
We have a couple of questions from the attendants on the online platform. So for the first one, she asks, what are the differences or pros and cons between lipid nanoparticle and the viral transfer in terms of gene therapy?
Yeah, I think that's still a question that needs to be answered. The big advantage of viral vectors is that we know that they can work. We've actually, demonstrated that, and in many ways we're taking advantage of millions of years of evolution. The disadvantages, is inflammation, as I mentioned, and the carrying capacity, because there are just many genes that will not fit into the AAV vectors. So I think one of the advantages of the lipid nanoparticles will be the larger carrying capacity. You have ability to engineer both of them, so I'm not going to say that one is better than the other. We've certainly engineered viral vectors in a number of ways, but lipid nanoparticles might be a little more flexible. in that way. So I'm not sure it's it's a little bit of an apples oranges, comparison. They have different things. So I don't know if lipid nanoparticles will ultimately be better, but if we can demonstrate that they have less inflammation, then perhaps, but at least on our initial attempt, we saw some inflammation. So there's still a lot of work to be done.
Thank you so much. And another question is what is the regulation that we should looking for the gene therapy? Should they get the insurance before the treatment also?
An insurance before treatment. Well, I mean, in terms of the regulations, every country has its own regulations, and in the United States, the FDA has pretty strict guidelines around gene therapy, in terms of the dividing, treating gene therapy, patients with gene therapy. I think in terms of insurance, that's a whole nother question. It's just because you can do something doesn't mean that someone's going to pay for it. And this has been a big, debate in the United States of will insurance, pay for gene therapy? And I think that we've been very lucky with Luxturna in that thus far the insurance companies have paid for it. And for patients who don't have insurance in the United States, Medicaid or kind of the socialized medicine has actually also agreed to pay for it. So I can't really answer whether or not you should get insurance, but I do think that we got lucky because this is the first time and there aren't many patients, and eventually insurance companies are going to say, we can't afford this. And so we do need to, find more cost-effective ways of treating these patients. Okay.
So yeah, I think that's the end of our
right. Thank you very much.
Q&A session. Yeah, once again, we'd like to say thank you to both of our speakers today, Dr. J. Timothy Stout and also Dr. Mark Pennesi, for such an interesting and inspiring talks today.
ค่ะขออนุญาตประชาสัมพันธ์นะคะจะมี QR code แบบประเมินความพึงพอใจในการสัมมนาในวันนี้นะคะรบกวนผู้เข้าร่วมการสัมมนานะคะช่วยทำแบบประเมินความพึงพอใจให้กับทางผู้จัดด้วยนะคะขอบคุณค่ะโดยสามารถสแกน QR code ด้านหน้าจอได้เลยนะคะค่ะสำหรับอาหารกลางวันนะคะจะวางอยู่ที่หน้าห้องประชุม110ทุกท่านสามารถหยิบที่ด้านหน้าห้องได้เลยค่ะขอบคุณค่ะ